Hey everyone, and welcome back to Gray's Gyan. I'm really excited about today's video because we are going deep inside the nuclear envelope. Now, before we jump in, I want to ask you something. Have you ever wondered, how does the nucleus know what to let in and what to let out? The nucleus holds your entire genetic code. It can't just leave the door wide open, right? It needs a system. A gated system. And that system is what we are going to study today. By the end of this video, you will understand the complete structure of the nuclear envelope, the proteins of the inner membrane, and why one mutation can destroy your muscles and heart, the nuclear lamina and the fascinating family of diseases called laminopathies, the nuclear pore complex, one of the most complex molecular machines in biology, how molecules travel in and out of the nucleus, and the RAN-GTPs, the master controller of all of that traffic. This content is straight from Gray's Anatomy and the Kirzenbaum and Trace Histology and Cell Biology textbook, so you can trust every word. Let's get into it. Before anything else, I want to show you this diagram. This is figure 1.18 directly from the textbook. I want you to get familiar with it because everything we discuss today is visible right here. On the left side, you have a beautiful diagrammatic cross-section of a nuclear pore complex. We call it the NPC. Let me walk you through what you're seeing. At the top is the cytoplasm. That's the outside of the nucleus. At the bottom is the nucleus itself. Running horizontally, you can see two membrane lines, the outer nuclear membrane, or ONM, and the inner nuclear membrane, or INM. The tiny gap between them is called the perinuclear space. Right in the middle, where the two membranes seem to pinch together, that is the nuclear pore complex itself. You can see the inner pore ring shown in blue, flanked by those golden yellow spheres. Those are the protein particles of the octagonal rings, one on the cytoplasmic side, one on the nuclear side. Coming down from the nuclear side, you see those converging lines. That funnel-like structure is the nuclear basket. It projects into the nucleus. On the cytoplasmic side, you see wavy lines going upward. Those are the cytoplasmic fibrils, protein fibrils associated with each protein particle. At the bottom left, you can see the nuclear lamina, that dense meshwork just beneath the INM. And on the right, it labels lamins A, B, and C. Those are the proteins that make up that meshwork. Now look at the right side of the figure. Panel A at the top. This is an actual electron microscopy image using a technique called freeze fracture. You are looking at the nuclear envelope from the top. Each of those circular ring structures you see, every single one of those is a nuclear pore complex. Look at how many there are. In an actively dividing cell, there can be up to 4,000 of these. Panel B at the bottom. This is a thin section side view. You can see the nuclear envelope as a horizontal band. The dark material you see on the nuclear side, that is condensed chromatin, or heterochromatin, that tends to cluster right at the nuclear envelope during interphase. You can also see the central pore channel labeled, that tiny gap where molecules pass through. Keep this image in your mind. We will refer back to it throughout the video. The nuclear envelope consists of two membranes, the inner nuclear membrane, or INM, and the outer nuclear membrane, or ONM, separated by a space of about 40 to 50 nanometers. That space is called the perinuclear space. Now here's something really important that students often miss. The nuclear envelope is not isolated. It is physically connected to the endoplasmic reticulum. Look at the diagram thumbnail on this slide. See how the outer nuclear membrane extends and merges with the endoplasmic reticulum? The perinuclear space is continuous with the endoplasmic reticulum lumen. They are literally one connected space. This means the outer nuclear membrane shares its membrane protein components with the endoplasmic reticulum. And because of that, just like the rough endoplasmic reticulum, you'll find ribosomes studding the outer surface of the ONM. Now the inner nuclear membrane is a different story. It has its own unique set of integral membrane proteins. Two crucial ones are lamin B receptor and emerin. Both of these provide binding sites for chromatin bridging proteins. We will look at these in detail in the next slide. One more thing to note, during interphase, which is when the cell is not dividing, condensed chromatin, heterochromatin, tends to pile up right against the inner face of the nuclear envelope. You saw it in the electron microscope image, but interestingly, at the sites of nuclear pores, chromatin is actually less condensed, which makes sense because those are the sites of active gene expression and RNA export. This slide is clinically important. Pay attention. The inner nuclear membrane has two key integral proteins that you absolutely need to know. Number 1, laminin B receptor. This protein anchors the nuclear lamina, specifically the laminin B proteins, to the inner nuclear membrane. It acts as a bridge between the structural meshwork inside the nucleus and the membrane itself. It is also involved in tethering heterochromatin. Number 2, emmerin. Emmerin provides binding sites for chromatin bridging proteins. It plays a role in holding the chromatin organization in place. Now here's where it gets clinically critical. A mutation in the gene encoding emmerin, the EMD gene, located on the X chromosome, causes Emery-Dreifuss muscular dystrophy, which we abbreviate as EDMD. What does EDMD look like? 3 main things. First, early joint contractures, particularly of the elbows, neck, and Achilles tendons. The joints get stiff and can't move through their full range. Second, skeletal muscle wasting, mainly in a humeroperineal distribution, meaning the upper arm and lower leg muscles. Third, and this is the most life-threatening, cardiomyopathy and cardiac conduction defects. Patients with EDMD can develop dangerous arrhythmias and sudden cardiac death. This is why these patients need close cardiac monitoring. Now, because the EMD gene is on the X chromosome, X-linked EDMD follows X-linked recessive inheritance. It primarily affects males. Females with one mutated copy are carriers. But here's the twist, and we'll come back to this. There is also an autosomal dominant form of EDMD. Same clinical picture. Different gene. That form is caused by mutations in laminin A, which brings us to our next slide. This is the most content-rich slide. Take your time. The nuclear lamina. Let me paint you a picture. Imagine the inside of a nuclear membrane, the inner face of the INM. Now imagine a dense, protein-rich meshwork lining that entire inner surface, like a chainmail coat for the nucleus. That is the nuclear lamina. It is 15 to 20 nanometers thick. The major components of this meshwork are proteins called lamins. Lamins are classified as type 5 intermediate filament proteins, so they are structurally similar to other cytoskeletal proteins like keratin and vimentin, but these ones are specifically inside the nucleus. There are 2 classes. A-type lamins, laminin A and laminin C, both encoded by the LMNA gene. B-type lamins, laminin B1 and laminin B2, encoded by separate genes. What does the nuclear lamina actually do? 3 major functions. 1. It mechanically reinforces the nuclear membrane. Without it, the nucleus would be soft and fragile. 2. It determines the shape of the nucleus. The characteristic round or oval shape of most nuclei depends on an intact lamina. 3. It provides binding sites for proteins that anchor chromatin to the cytoskeleton. Now, here's a striking fact. Laminin A is the most mutated protein linked to human disease. Over 350 disease-causing mutations have been identified in the LMNA gene. These diseases are collectively called laminopathies. Let's look at the major ones. First, Hutchinson-Gilford progeria syndrome. This is caused by a specific mutation in the LMNA gene that produces a truncated, abnormal form of laminin A called progerin. Progerin gets stuck at the inner nuclear membrane and causes mechanical stress, DNA damage, and accelerated cell senescence, particularly in mesenchymal stem cells and vascular smooth muscle cells. The result? These children display premature cardiovascular disease. They suffer heart attacks and strokes as young children and typically die in their early teens. It is a devastating condition. Second, autosomal dominant EDMD. As I mentioned earlier, different mutations in LMNA also cause EDMD. Same disease picture, contractures, muscle wasting, cardiomyopathy, but this time it's autosomal dominant, and the culprit is laminin A, not emmerin. This is important to distinguish in exams. Third, autosomal dominant leukodystrophy. This one is caused not by mutations in lemon genes, but by overexpression of lemon B1. Too much of a good thing. The result is progressive demyelination in the central nervous system. The myelin sheaths around nerve fibers break down gradually, leading to a slow and progressive neurological decline. And here's some fascinating animal model data from the textbook. Mice lacking both lemon B1 and lemon B2 survive until birth, but their neuronal development is severely compromised. This demonstrates that lemon B proteins are absolutely essential for brain development. Knock them out, and the brain doesn't form properly. The key references for this section are Warman 2012, Burke and Stewart 2013, and the most recent update, Pandey and Ghosh 2023, all cited in Gray's Anatomy. Let's now talk about one of the most sophisticated molecular machines in all of biology, the nuclear pore complex, or NPC. Look at the electron microscopy images on the left. At the top, that's the freeze fracture view from Figure 1.18a. You can see individual pores from above, each appearing as a ring embedded in the membrane. At the bottom, the thin section side view from Figure 1.18b, you can see the nuclear envelope in cross-section, with the condensed chromatin sitting right below it. Now let's talk numbers, because these numbers are impressive and they are exam favorites. The NPC has an outer diameter of 130 nanometers. That is large for a protein complex, large enough to be visible by electron microscopy. The inner pore, the central channel, has an effective diameter of only 9 nanometers for free diffusion. Anything small enough to fit through that 9 nanometers channel can pass freely. This includes small ions, water molecules, metabolites, and proteins up to approximately 17 kilodaltons. Each NPC is built from more than 50 different proteins called nucleoperins, or NUPs. That is an enormous number of proteins for a single structure. An actively transcribing cell can contain up to 4,000 nuclear pore complexes in its nuclear envelope. Think about that. 4,000 of these giant machines, all working simultaneously. And structurally, the NPC has beautiful eightfold rotational symmetry. It is shaped like an octagon when viewed from above, which is exactly what you see in the electron micrograph. The key structural elements, as labeled in Figure 1.18, are the cytoplasmic octagonal ring, a ring of protein particles on the cytoplasmic face. The nuclear octagonal ring, same structure on the nuclear face. The inner pore ring, which spans through both the INM and ONM, holding everything together. The nuclear basket, a funnel-like structure projecting into the nucleus. The cytoplasmic fibrils. Protein fibrils extending into the cytoplasm. And critically, the INM and ONM fuse around each pore complex. They bend toward each other and merge to create a continuous channel from one side to the other. The key research references here are Races and D'Angelo 2012, who showed that NPC composition actually varies between different cell types, and Petrovich et al. 2022, who used cryo-electron microscopy to resolve the NPC structure at near-atomic resolution. Incredible work. Alright. We know what the pore looks like. Now let's understand what it does. The nuclear pore complex acts as a highly selective, directional molecular filter. Think of it like a customs checkpoint at an international airport. Things can go in, things can go out, but they need the right credentials, and they move in specific directions only. Let's start with import. What goes into the nucleus? The nucleus needs histones for packaging DNA, transcription factors and gene regulatory proteins for controlling which genes are switched on, DNA and RNA polymerases for replication and transcription, and various signaling molecules. All of these are made in the cytoplasm and need to be transported into the nucleus. How? Most large proteins enter the nucleus as complexes with specific carrier proteins called importans. Here's the sequence. Step 1. The cargo protein carries a short sequence of amino acids called a nuclear localization sequence, or NLS. This is basically its entry visa. It marks the protein as something that belongs in the nucleus. Step 2. The NLS binds to an importin, either directly or through an adapter protein. Step 3. the important cargo complex moves through the NPC. Importantly, this translocation is energy independent. It doesn't require ATP at this step. Step 4. Once inside the nucleus, the cargo is released. The important then shuttles back to the cytoplasm, ready to carry the next cargo. Now let's look at export. What comes out of the nucleus? The nucleus constantly produces mRNA, which must get to ribosomes for translation, tRNA, for the translation machinery, and ribosomal subunits, assembled in the nucleolus. These are carried out by complementary carrier proteins called exportins. The process is the mirror image. Cargo binds exportin inside the nucleus, the complex moves through the NPC, and cargo is released in the cytoplasm. The exportin then returns to the nucleus for reuse. And for small molecules, ions, metabolites, proteins under about 17 kilodaltons, none of this ceremony is needed. They simply diffuse freely through the 9 nanometers central channel. So to summarize, importins bring things in, exportins take things out, and small stuff moves freely. But what provides the directionality? What makes sure importins go the right way? That is where our next topic comes in. This slide is conceptually important. Slow down and build the analogy. The master regulator of all nuclear transport is a small protein called RAN, which stands for RAS-related nuclear protein. RAN is a GTPase, meaning it can hold either GTP, the active form, or GDP, the inactive form, and it can hydrolyze GTP to GDP. Here's the key concept. RAN creates a concentration gradient across the nuclear envelope. Look at the 2 boxes on this slide. Inside the nucleus, RAN is predominantly in the GTP-bound form. RAN GTP is high. Why? Because inside the nucleus there is an enzyme called RCC1, the RAN-GF, guanine nucleotide exchange factor. RCC1 is bound to chromatin and constantly converts RAN-GDP into RAN-GTP inside the nucleus. In the cytoplasm, RAN is predominantly in the GDP-bound form. RAN-GDP is high. Why? Because in the cytoplasm there is a RAN gap, GTPase activating protein, which converts RAN-GTP back into RAN-GDP. This creates a steep gradient, RAN-GTP high in nucleus, RAN-GDP high in cytoplasm. Now, why does this matter? Because RAN-GTP and RAN-GDP behave differently when they interact with importins and exportins. When an important cargo complex enters the nucleus, it encounters RAN-GTP. RAN-GTP binds the importin and causes it to release its cargo inside the nucleus. Perfect, the protein has been delivered where it needs to go. When an exportin cargo complex leaves the nucleus and reaches the cytoplasm, the RAN-GTP on board gets converted to RAN-GDP by RAN gap. This change in RAN causes the exportin to release its cargo in the cytoplasm. Perfect delivery again. Think of it this way. RAN-GTP is like a VIP badge that only works inside the nucleus. When you have that badge, cargo gets released from importins and loaded onto exportins. When you leave the nucleus, your badge expires, cargo falls off the exportin into the cytoplasm. This gradient is completely self-maintaining. As long as the cell keeps RCC1 in the nucleus and RAN gap in the cytoplasm, the system runs continuously. The key references are Clark and John 2008, and Races and D'Angelo 2012. We've talked about the nuclear envelope in the context of an interface cell, a cell that is not dividing. But what happens when the cell decides to divide? First, a quick note on chromatin organization during interface. As we mentioned earlier, condensed chromatin, heterochromatin, aggregates at the nuclear periphery, right against the inner face of the nuclear envelope. You can see this beautifully in the electron microscope image on this slide. Those dark regions are the heterochromatin. But notice that the chromatin is less condensed at the nuclear pores. This allows the active genes in those regions to be transcribed and their RNA exported through the nearby pores. Very efficient organization. Now, cell division, specifically mitosis and meiosis. The nucleus faces a major challenge. To divide the chromosomes, you need to completely dismantle the nuclear envelope and then rebuild it around the daughter nuclei. Here's how that happens step by step. Step 1. Prophase. The cell signals the beginning of mitosis, and a cascade of kinase activity leads to phosphorylation of the lamin proteins. When lamins are phosphorylated, their filament structure disassembles. Without the lamina scaffold, the nuclear membranes begin to vesiculate. They break up into small membrane vesicles that disperse into and merge with the endoplasmic reticulum. The nuclear envelope ceases to exist as a distinct structure. Step 2. Metaphase and anaphase. The chromosomes are now free in the cytoplasm, lined up on the mitotic spindle. There is no nuclear boundary. The nuclear envelope proteins are temporarily stored in endoplasmic reticulum membranes and vesicles throughout the cytoplasm. Step 3. Telophase. As the chromosomes reach the poles and begin to decondense, nuclear periphery proteins, including the lamins, begin to reassociate with the surfaces of the chromosomes. This happens even before the membranes return. These proteins provide docking sites for the membrane vesicles. Step 4. Reassembly. The membrane vesicles fuse together around the chromosomes, reconstituting the nuclear membranes. Then the lamins dephosphorylate, remove their phosphate groups, which allows them to polymerize back into filaments, rebuilding the nuclear lamina. The nuclear envelope is complete again. This is why laminopathies are so destructive. Every time a cell divides, the nuclear envelope must be rebuilt correctly. If the lamins are defective, the rebuilding is faulty. The nucleus becomes structurally weak, prone to mechanical damage, and accelerated death. References: Simon and Wilson 2011, Miyamoto and Harada 2021. I want to take a moment to acknowledge the researchers whose work underpins everything we have discussed today, because Gray's Anatomy specifically cites these papers, and as medical and biology students, it's important to know who discovered what you're learning. Warman, 2012, published in the Journal of Clinical Investigation. This is the foundational comprehensive review of lamin biology and the full spectrum of laminopathy diseases. If you want one paper to read on laminopathies, this is it. Burke and Stewart, 2013, focused specifically on lamin A and progerin. Showed in detail how progerin disrupts nuclear architecture and causes DNA damage in vascular smooth muscle cells, which is what drives the cardiovascular disease in progeria patients. Pandey and Ghosh, 2023, the most recent review in this field, updating our understanding of laminopathy mechanisms and therapeutic possibilities, including gene therapy approaches for EDMD and progeria. Races and D'Angelo, 2012, published in Nature Reviews Molecular Cell Biology. This paper challenged the idea that all nuclear pore complexes are identical, showing instead that NPC composition varies between different cell types and developmental stages. Petrovich et al., 2022. A landmark cryo-electron microscopy study that revealed the near-atomic structure of the NPC. This is like going from a blurry photograph to a crystal-clear high-resolution image of a machine we've been trying to understand for decades. Clark and Zhang, 2008, in Biochemical Journal. The mechanistic study of the RAN-GTPase pathway, explaining how the RAN gradient is generated and maintained, and how it provides directionality to all nucleocytoplasmic transport. Simon and Wilson 2011, published in Nature Reviews Molecular Cell Biology, Volume 12. They described how the metazoan nucleoskeleton, a poorly understood internal scaffold inside the nucleus, includes nuclear pore-linked filaments, A-type and B-type lamin intermediate filaments, nuclear mitotic apparatus, NUMA, networks, spectrins, titin, unconventional polymers of actin, and at least 10 different myosin and kinesin motors. Miyamoto and Harada 2021, The Journal of Biochemistry, Volume 169. They argued that, while it had been believed that the dense nuclear environment prevents the formation of a cytoskeleton, like network of protein filaments, accumulating evidence suggests that the cell nucleus also possesses structural filamentous components to support nuclear organization and compartments, referred to as nucleoskeleton proteins. All of these are excellent further reading if you want to go beyond the textbook. Alright, let's bring it all together. Point 1. Structure. The nuclear envelope is a double membrane, inner nuclear membrane plus outer nuclear membrane, separated by a 40 to 50 nanometer paranuclear space. The ONM is an extension of the ER. The INM has its own special proteins, emerin and lamin B receptor. Point 2. Nuclear lamina. A 15 to 20 nanometer meshwork of lamin proteins, A-type and B-type, on the inner face of the INM. It shapes the nucleus, reinforces it mechanically, and anchors chromatin. Over 350 mutations in lamin A alone cause the laminopathies. Point 3. Nuclear pores. 130 nanometers outer diameter. 9 nanometer inner channel. More than 50 nucleoperin proteins. Up to 4,000 per active cell. 8-fold octagonal symmetry. Free passage for molecules under 17 kilodaltons. Point 4. Transport. Importins carry NLS-tagged proteins into the nucleus. Exportins carry RNA and other molecules out. Small molecules pass freely. The RAN-GTP or GDP gradient across the nuclear envelope provides the directionality. Now, for those of you who need a memory aid, here is a mnemonic I put together for laminopathies. Think of the word lemon. L. lamins. A. B1, B2, C. The structural proteins of the nuclear lamina. E. emerin. Mutated in X-linked EDMD. M. Mutations. Over 350 in lamin A, the most mutated human disease protein. O. Onset early. Progeria causes premature aging and death in the teens. N. Neuronal. Lamin B is essential for neuronal and brain development. Lemon. Write it down. It will save you in your exams. And that's a wrap on the nuclear envelope. If this video helped you, please do me a favor. Hit the like button, it genuinely helps this channel grow. And if you are not subscribed yet, click subscribe so you don't miss the next video. Drop a comment below, let me know which part was most interesting to you, or if you have any questions. I read every comment. Keep learning, keep asking questions, and I will see you in the next video.